Today I'll be discussing the structural organization of proteins as part of your revision. The co-house question which has come is they have asked a 10-mark question on the structural organization of proteins and to name the bonds which stabilize protein structure. Or you may get one of the structural forms, that is the secondary level or the tertiary level or the quaternary level. Any of these can come as a 3-mark or a 5-mark question. Now, if you are asked as a 10-mark question, the structural organization of proteins, what would you write? Firstly, you have to say that conformation or the structural organization of the protein is very important for its function. If a protein is existing as a stretched out polypeptide chain, it does not have any biological activity. So what is conformation? Conformation is the three-dimensional arrangement of the atoms or the amino acids in a structure. And for this, it is important to know the amino acid sequence because they specify the conformation. After that, after the short introduction, you can say that there are 4 levels of organization. One is the primary, second is the secondary, tertiary, and quaternary. Next we go on to discuss each level of conformation. The first being primary. Define what is primary structure. Primary structure is the sequence of amino acids in a protein. Okay, it is just the sequence of amino acids in a protein. Secondly, why is it important? Why is this primary structure important? Because many genetic diseases, they result in proteins with abnormal amino acid sequences. If the amino acid sequences are abnormal, that is, there is a mutation or a difference in the amino acid sequences, many genetic diseases are a result of that. Secondly, these abnormal amino acid sequences can cause improper folding. When there is improper folding, what happens? It loses its normal function. Okay. Next we talk about the fact that the amino acids in the primary structure are linked to each other by the peptide bond. You may be asked separately as a 2-mark or a 3-mark question on the characteristics of a peptide bond. So what will you write about the characteristics of a peptide bond? The peptide bond has got a partial double bond character. That is, it's shorter than a single bond but longer than the double bond. What is the bond length? It is 1.32 angstrom units. The single bond length is 1.49 angstrom units and the double bond length is 1.27 angstrom units. So it is shorter than a single bond and longer than a double bond. The peptide bond is rigid and planar. That is, there is no rotation between the carbonyl carbon and the nitrogen of the peptide bond. And the peptide bond, you can see the groups, the double O and the NH are in trans position. So it's a trans bond. And it is uncharged but polar in nature. So that is the importance of the primary structure. That's what you need to write for the primary structure. Now, what would you write about the secondary structure? The secondary structure has been described by Pauling and Corey, and it refers to the spatial arrangement of amino acid residues that are near one another in the linear sequence. It is of 2 types. You have alpha helix and the beta pleated sheet. So first you discuss the alpha helix. What can you discuss about the alpha helix? With figure, that is important. The alpha helix has a spiral structure. Okay, the amino acids are tightly packed or coiled along the polypeptide backbone. Now you find that the side chains or the R groups are extending outwards from the central axis, and this is to avoid steric hindrance. Give some examples like keratin, a fibrous protein which contains nearly the entirely the alpha helical structure, and this is mainly seen in the hair and skin, and it's a rigid structure. Another example is myoglobin. It is also highly alpha helic, but its structure is globular and it is more flexible. Now you have to give the features of the alpha helix. Okay, so what are the features of the alpha helix? What type of bond do you see in the alpha helix? It's only the hydrogen bond. So the hydrogen bonds are intrachain. These intrachain hydrogen bonds are running parallel to the spiral or the central axis, and it is formed between the peptidyl bond carbonyl oxygen, the oxygen of the carbonyl group of the peptide bond, and the amide hydrogen, as you can see in the figure. These hydrogen bonds are formed between the peptide bond carbonyl oxygen and the amide hydrogen. These hydrogen bonds are weak, but collectively they tend to stabilize the helix. Then you have to go on to describe in detail the helix. So there are 3.6 amino acids per turn of the helix. There are certain amino acids which can disrupt the helix, like glycine, proline, charged amino acids, or amino acids with bulky side chains. The pitch is 0.54 nanometers. That is, one turn of the helix covers a distance of 0.54 nanometers, and the distance between each residue is 0.15 nanometers. The helix may be either right-handed or left-handed. So these are all the details you have to give about the alpha helical structure, okay, that is, which is part of the secondary structure of the protein molecule. The next type of secondary structure is the beta pleated sheet. So in the beta pleated sheet there may be two or more peptide chains. They are called beta strands, and they become the segments of the polypeptide chain. They are almost fully extended. Now here the hydrogen bonds are not running parallel but perpendicular to the backbone, and such beta pleated sheets are seen in globular proteins, and almost all of the peptide bond are involved in hydrogen bonding here. Now to describe the beta pleated sheet still more, you have to say that the polypeptide chains may be arranged either anti-parallel, that is, the N-terminal and the C-terminal will alternate with each other, or they run parallel, that is, the two N-termini are lying adjacent to each other. The hydrogen bonds formed between separate polypeptide chains are called interchain bonds, and those within the same polypeptide chain is known as the intrachain bonds. Tertiary structure. What is the tertiary structure? The tertiary structure refers to the three-dimensional structure of a single protein molecule. So here what do you see? The alpha helix and the beta pleated sheets are folded to a compact globular protein. The hydrophobic side chains are buried in the interior, whereas the hydrophilic groups are generally found on the surface of the molecule. This structure shows the lowest energy, the greatest stability state of a polypeptide chain. Now you can be asked, what are the bonds which hold the protein structure, particularly the tertiary and the quaternary structures? So what are the bonds stabilizing the structure? So the unique three-dimensional structure of the polypeptide chain, as I told you, is determined by its amino acid sequence. The interaction between the amino acid side chains guide the protein or the polypeptide chain to fold in a compact structure. Okay, there are four types of interactions which cooperate in stabilizing the tertiary structure. Now let's look at these different types of interactions. Now first we have the disulfide bond. This disulfide bond is a covalent linkage formed between two cysteine residues. The cysteine residues may be separated from each other by many amino acid residues, or it may be located on different polypeptide chains. The folding will bring them in close proximity, resulting in the formation of a covalent bond. This disulfide bond stabilizes the three-dimension structure and resists it from getting denatured. So first is the disulfide bond. Now, I had earlier told you in the secondary structure there is the hydrogen bonds. So the amino acid side chains containing oxygen like serine or threonine, or the nitrogen bound to hydrogen, can form hydrogen bonds with electron-rich atoms like the oxygen of a carboxyl group or the carbonyl group of the peptide bond. So formation of such hydrogen bonds between polar groups on the surface of the proteins with the aqueous solvent enhances the stability of the protein molecule. So these two are covalent interactions, that is the disulfide bond and the hydrogen bond are covalent interactions. Now we come to those which are non-covalent interactions. That is, one is the hydrophobic interaction. So amino acids with non-polar side chains, they tend to be located towards the interior of the polypeptide chain, and they associate with similar hydrophobic amino acids. Now what happens to the amino acids with polar or charged side chains? They are located on the surface in contact with the polar solvents. So towards the interior of the polypeptide chain, you find the amino acids with non-polar residues like in the case of isoleucine, valine, tryptophan, and they associate with similar hydrophobic amino acids like alanine, phenylalanine and leucine. So they are all found associated with each other towards the center or the interior of the polypeptide chain. Next is the ionic interaction. Negatively charged groups such as those amino acids which contain acidic group like the COO group in the side chains of aspartic acid or glutamic acid will interact with positively charged groups like the amino group you see in the side chains of those basic amino acids like lysine or arginine. So the negatively charged acidic group, the COO minus group in glutamic acid or aspartic acid, will form an ionic interaction with the positively charged group, that is the basic amino group of side chains of lysine and arginine. And lastly, the fifth is the van der Waals interaction. So this van der Waals interaction is a weak, non-specific attractive force created when two atoms approach each other. So when these two atoms approach each other, then there is a transient electric dipole which is formed between the two atoms, which attracts each other weakly. And this is known as the van der Waals interaction. The next level of organization is the quaternary structure. So proteins with a single polypeptide chain is called monomeric. When it has more than one polypeptide chain, it is called as dimeric or tetrameric or whatever it may be. Okay. Now these polypeptide chains may be identical to each other when they are known as homopolymers, and when they are not identical to each other, it is known as heteropolymers. And the arrangement of these polypeptide subunit is called as the quaternary of proteins. So each subunit is held by non-covalent interactions. What are these non-covalent interactions? Like the ionic bond, the hydrophobic bond, etc. And they may function independently or cooperatively. Okay. For example, in the case of hemoglobin, there is a cooperation. It is acting cooperatively. That is, the binding of one oxygen molecule to one of the subunits is facilitating the binding of oxygen to the other subunits. So this finishes our discussion on the structural organization of proteins. So please remember, for a 10-mark question, you should be able to write in detail about the primary, the secondary, the tertiary and the quaternary structures. You should give in detail what is secondary structure, types of secondary structure, and the details about the alpha helix and the beta pleated sheet. In the case of tertiary and quaternary structures, you should be able to describe the interactions which hold the structure together, both the non-covalent and the covalent interactions. Okay. Secondary structure, if you are asked a 5-mark or a 3-mark question, you should be able to write both in detail about the alpha helix and the beta pleated sheets. Draw diagrams wherever necessary. That is very